Hello, I'm Dr. K.S. Jyoti. I'm an assistant professor at the Department of Biotechnology in Telangana Mahila Vishwavidyalaya. Today we will talk about a very interesting topic, identification of bacteria. Let's start understanding all the basics of why identify bacteria and what are the different ways in which we can identify bacteria and what are the important techniques involved in this. So our topic of discussion in this session will be about identification of bacteria using staining techniques and some biochemical tests that are also important for the identification. Now, the first very important question is, what are the situations where we will have to identify bacteria? Now, technically, we know that bacteria are microorganisms. Now, there are good bacteria, bad bacteria. There are certain bacteria which cause a lot of diseases. So whenever you're talking about laboratory tests where you want to check if a particular sample is infected with a bacteria or no, or for example, let's say we took a soil sample, you took a water sample, and you want to check if there is any bacteria or a microorganism that is present in that particular sample, any kind of a sample analysis that you're doing, it is in these cases that we have to identify the presence of the bacteria. Now, there are various ways in which we can know, but suppose you want to study and these are microorganisms, so the only way we will be able to study them is using a microscopy. Now, when you want to visualize a bacteria under a microscope, because the bacteria are colorless, you need to add some colored stain so that you will be able to visualize. Now, the whole idea is we want to create a contrast between the organism and the background. So when you add some color, if the bacteria take up this color and the background is in an opposite color, then you will be able to visualize bacteria under the microscope. So technically, you have to add a stain so that you can identify bacteria under a microscope. So in any routine laboratory test sample analysis also, whether you're taking a blood sample, urine sample, water sample, stool sample, what they do is they first check if the sample is contaminated with any bacteria or no, for which they will have to add this stain. Now, once you get to know that there is a bacterial contamination or an infection in that particular sample, further what we should do, we should identify what type of a bacteria is it. So we all know, we must have studied the classification of microorganism, classification of bacteria. Different types are there. So we should know what type of a bacteria is it. We should study their biochemical characteristics, structural properties, starting from the simple characteristic features like knowing about its cell wall structure, the size of the bacteria, the arrangement of the bacteria. Now, starting from this to the highly complicated things like the nature of the bacteria and the structural details, biochemical details, all these things can be evaluated with the staining techniques and further with the biochemical tests. So knowing what type of a bacteria is present in a particular sample is very important diagnostically or in terms of your water samples also, you need to analyze all these things. So identifying bacteria using staining techniques is a very age-old process. There are various techniques that have been identified for this. So what are the prominent types of staining? One, you have simple staining, differential staining, and in some very special cases, if you know that the bacteria has any specific characters like spore formation or flagella in their structures, you can also use a special staining. So let's start talking about simple staining. As the name indicates, why is this called as a simple staining? Both the things. One, it only uses one type of a stain, and even the process that or the procedure that is used is very simple. It's just a 3-step process. Now, what happens in simple staining? We use either of these colored dyes like a methylene blue, crystal violet, or a saffronin. Just take the bacterial sample, make it as a smear, and then you stain it with any of these colored dyes. Now, all the bacteria which take up this color will be visualized like this, as you see in the picture. in the image on the screen. This is how you will be able to visualize the bacteria. But what happens in a simple staining? All the bacteria will appear in the same color as the stain. Because here we can only understand the morphology. Like I said, you will be able to study the size, arrangement, and shape. That's it. You will not be able to identify anything else about the bacteria in a simple stain. Now, all these colored stains that you're using in the technique, methylene blue, crystal violet, saffron, basically they all have a kind of positive charge, which will bind to the negative charge of the bacterial cell wall, whether it is positive or negative bacteria, any class they will get. So all bacteria will have just one color. Let's say I take methylene blue, all the bacteria you see here stained with methylene blue, they appear blue in color. That is a simple staining. Now, this is a very basic technique. So this simple staining sometimes is also called as a direct staining. So where do I use a technique like a simple staining or a direct staining? Only in the first, I just get a sample and I want to know whether if this has any bacterial contamination or no. So as a first step, I will do simple staining. Suppose the sample I got tests positive and I know that there is some bacterial infection. Then further what I should do, I should continue the process, do a differential staining where I want to distinguish between the kinds of bacteria that are present in the sample. So simple staining, also called as direct staining. I can use any colored dye which is positively charged. And what is the information I will get from a simple stain? First, presence or absence of bacteria. And then I will be able to know the size, shape, and arrangement of the bacteria. That's it. I will only able to know about the morphological features, nothing about the characters or behavior of the bacteria using a simple staining. A very important advanced method of staining that is prominently used in all diagnostic laboratories for research purpose is called as a differential staining. Now, the difference between this is in differential staining, you will use more than one type of a stain. And the additional information of the bacteria, other than what is its cell size, shape, everything, you will be able to get that information from a differential staining. Here, we can differentiate them also based on their chemical or structural properties. Mostly when we talk about the structural properties, we would say the cell wall structure of the bacteria. So based upon the cell wall structure, we know bacteria can be divided into gram positive, gram negative. So all those differentiations can be studied using a differential staining. That is why the name itself is differential staining, where you can differentiate them based upon their characteristic features, and you will be able to study them in detail. So what are the prominent types of differential staining? Gram staining, acid-fast staining, endospore staining. These are the three prominent types of differential staining, out of which we regularly use gram staining and acid-fast staining. When it comes to research purpose, when you're using it in for a study purpose of a bacteria, we generally use gram staining. Diagnostically, if you want to know particularly if the bacteria, if the sample has a mycobacterium infection or no, then we use an acid-fast stain. So the most prominent staining technique that is popularly used in any laboratory while you're learning also, we study widely about this. This is called as gram staining. Now, a very innovative method that is identified by a Danish microbiologist or a bacteriologist is Christian Gram. He identified this way back in 18th century. Now, it provides all important information about the bacteria. So if you can see in the screen, you will see this is how the bacteria gets stained differently. Gram positive bacteria, you will see, will get purple colored, whereas gram negative bacteria will stain pink. Also, you will see that the bacteria is rod shaped and this bacteria is cocci. So whether you will be able to identify the shape very clearly, circular bacteria, rod-shaped bacteria, that differentiation also can be clearly visualized because it is differentially colored, different colors. So gram-positive colors purple, gram-negative bacteria will color pink, right? So how are we able to get these differences? As I said, this is prominently because of the differences in the cell wall composition, based on which you can divide bacteria into gram-negative and gram-positive. So all those bacteria that do not retain the stain, that is, they do not retain the Gram's stain, you call them as gram negative. All those that can retain are called as gram positive. So those that do not retain gram stain, we call them as gram negative. So the important understanding we need to have is why do they retain the stain and or why do they not retain the stain. So to understand this, it is very important for us to study the cell wall structure. Now, if you look at certain bacteria, in their cell wall, they have a very thick peptidoglycan layer. Peptidoglycan layer, very important structure in the bacterial cell wall. Peptidoglycan is a combination of peptides plus glycosaminoglycans or glycans, a part of carbohydrates, that is sugars. Now, this peptidoglycan layer on the surface, they have more complex sugar structures, which have a lot of amino sugars, which we call them as teichoic acids. So, you talk about gram-positive cell wall, they have a thick peptidoglycan layer with teichoic acids on the surface. Whereas gram-negative cell wall, they will have a very thin peptidoglycan layer, but the outer surface of this peptidoglycan layer has a lot of lipopolysaccharides present. Lipopolysaccharides are to the glycan of this peptidoglycan layer, that is the sugars, you will have lipid molecules attached. So this lipopolysaccharides or the LPS layer is very strong. It creates an outer membrane in which you have porins. These are proteins that will help the movement of the particles into and out of it. Right? So the important understanding is gram-positive cell walls will have a thick peptidoglycan layer and they'll have teichoic acids on the surface of the peptidoglycan layer. Gram-negative, they have a very thin peptidoglycan layer and they have a strong lipopolysaccharide layer. So quick differentiation, peptidoglycan layer is present in gram-positive, absent in gram-negative. Teichoic acids are present in gram-positive, absent. Whereas outer membrane containing lipopolysaccharides is absent in gram-positive. Now it is present in gram-negative. Now, what is our understanding here? In a gram staining process, just a second. Right. So what happens while we are staining the bacteria using gram staining procedure, and why do they retain the color? Now, what happens here is in the gram-negative bacteria, they have a very thick lipopolysaccharide layer. So in the treatment after adding the gram staining, when you treat it with alcohol, that alcohol goes into the lipopolysaccharide layers. So therefore, the stain is discolorized. The stain gets decolorized. And when we add a counter stain like safranin, that's a counter stain that is added. So that lipopolysaccharide layer takes up the counter color instead of the gram staining color that is added. But what happens in a gram positive layer, because they have a thick peptidoglycan layer, the color gets retained in this thick layer. Therefore, gram positive bacteria will appear purple color, whereas gram negative bacteria will appear pink in color. That is the difference. Okay. So gram positive bacteria, because of their thick peptidoglycan layer, they will retain the color. Whereas gram negative bacteria, because they have this lipopolysaccharide layer, they don't retain the gram stain color, but instead they take up the safranin color. So they either will look pink or red in color, whereas gram positive bacteria will look either purple or blue in color. So we know a lot of microorganisms that are there, many of them which are disease-causing microorganisms also. So you will see that most of these, that is Staphylococcus, Streptococcus, Corynebacterium, Clostridium, Listeria, they all belong to the gram-positive category, whereas most of your disease-causing organisms like E. coli, Pseudomonas, gonorrhea, Neisseria gonorrhea, all these belong to the gram-negative group of bacteria. So when you are doing a gram staining, what is the procedure? We will follow 4 simple steps in this. First, we put a primary stain, that is your crystal violet. Then you add a mordant, that is your grams iodine. Now, what we do is you add an acid or an alcohol to decolorize this, or sometimes we add a mixture of both ethanol and acetone. Now, what happens while you decolorize in gram-negative bacteria, because the lipopolysaccharide layer opens up, it allows this decolorization, removes this. But whereas gram-positive bacteria doesn't get affected with this decolorization, and it retains the gram stain. So when you counter stain with safranin, as I discussed already, gram-negative bacteria will take up the safranin color instead of the primary stain color. Therefore, gram-negative bacteria will appear red, whereas gram-positive will appear blue in color. So this is how you will see. First, you do the fixation step where you fix the bacteria onto the slide. Then you will add crystal violet, which is the primary stain, iodine, which is a mordant. Then you do an alcohol wash. The purpose of this wash is decolorization. It is in this step that you will see that the gram-positive bacteria will still retain the same color because the alcohol wash doesn't affect the peptidoglycan layer, whereas in gram-negative bacteria, it gets decolorized. Later, when we add a counter stain in gram-positive, because already this stain is retained, they don't take up this color. But whereas gram-negative bacteria, because they are decolorized after the alcohol wash, they will take up the counter stain and they will look red in color. So this is why, based upon their cell wall structure, they will take up different stains. That is the reason why we are calling this differential staining. Based upon their morphological, physiological characteristic features of the cell wall, you are able to differentiate and then observe whether the bacteria that you have in the sample is gram-positive or gram-negative. So what do we observe from the gram staining? Basically, if the gram staining is positive, I mean any color of stain, it will give us the information. First information that we will get is about the type of the bacteria. I will get to know whether it is gram positive or gram negative. Then shape of the bacteria. We saw in the image earlier that there are cocci and there are bacilli, rod-shaped structures or round-shaped structures you can identify. You can also identify the other characteristic features like the size, relative quantity, how many bacteria are there, and how are they arranged? I mean, are they arranged in groups? Are they present as individual bacterial colonies? We will be able to get to know all these features also if it relates to that particular bacteria. Now, if there are intracellular bacteria, if there is any bacteria that are present inside some other cells, or if there is presence of red blood cells, white blood cells, all these things also I will get to know from the gram staining results. Then if the gram stain sometimes can also identify fungi, if they are in the form of yeasts or molds also, I will be able to get. But of course, if you identify that it is a fungi or a yeast, then you will have to do further testings to check this. Okay? So the first type of differential staining that we understood is gram staining. Very important. Now the next type of staining is an acid-fast staining. Now sometimes what happens is, specifically, this is related to the bacteria that belongs to the genus Mycobacterium. That is those bacteria that are prominent in causing tuberculosis. So basically, when we get a sample, when we want to rule out whether that particular patient is positive for tuberculosis or no, instead of, I mean, first, let's say we do a primary staining, gram staining, we identify. Then when we want to confirm whether that particular sample is positive for tuberculosis or no, we can do this particular acid-fast staining. Now, what happens is, in this kind of a bacteria, Mycobacterium, they have a very thick, impenetrable cell wall structure because they have mycolic acid in them. Now, this bacteria belonging to Mycobacterium or Nocardia have an impenetrable cell wall structure. So your gram stain may not be able to give a clear idea about this. So therefore, we use a different type of a staining technique, what we call it as an acid-fast staining technique. Now, specifically, this acid-fast organisms, they resist the decolorization when we add an acid alcohol, and all those that resist the acid-fast organisms will appear blue in color. Those bacteria which could not sustain this, they will look pink in color. So we take a counter stain. So all those that retain, same thing. So first you add a primary stain, that is a carbonyl fusion. Then you give a heat treatment, moderate heat treatment. You give an acid alcohol wash to decolorize it. Now acid fast bacteria only will sustain this wash. They will not get decolorized. Therefore, they will retain the pink color. Whereas the non-acid fast bacteria, because they will get decolorized and they take up this counter stain that is methylene blue, they will look blue in color. So all acid fast bacteria will look pink in color. Non-acid fast bacteria will look blue in color. This is because acid fast bacteria do not get affected with this decolorization step. They will still be able to hold the color even after a wash with acid. That is why we call them as acid fast bacteria. So only the bacteria belonging to the species of Mycobacterium will belong to this acid fast bacteria. All other, like your E. coli, is a non-acid fast bacteria. So when you are in a doubt whether that particular bacteria is E. coli or any other organism causing the disease, to rule out that, you can do an acid fast staining, which will confirm whether the infection is caused because of the tuberculosis bacterium or of E. coli. So that is why this is also a type of differential staining. Now, let's say we have ruled out what type of a bacteria, we did the staining, we observed, but sometimes if certain bacteria have any specialized features, like they're able to form endospores or they have some flagellar-like structures, there are two other types of specialized staining techniques. Many are there. Some prominent ones are endospore staining, where you can visualize the bacterial endospores in certain species like Bacillus or Clostridium. They can resist staining with basic stains, but they stain very well with malachite green or safranin. Then certain types, you can do a negative staining. Some bacteria which have a polysaccharide capsules or slime layers, they will simply take the outstain. I mean, only the background is stained and the cells without with the capsule don't take up any stain. That is negative staining. So the background is strongly colored. Bacteria doesn't have any stain. So like this, with a negative staining also, you will be able to identify bacteria which are having these capsulated structures. So these fall under specialized staining. Now, we have done the staining. We identified that the sample has a bacteria, whether it is a gram negative or gram positive. But from staining, I will not be able to name or label what bacteria it is. Only in acid fast, at least because it is only one or two species that are sensitive to that, I will be able to know. But otherwise, I cannot label that this particular sample is affected with this bacteria. So further, I have to do biochemical tests, which will help us identify the exact genre of the bacteria that is causing the infection. Now, what is the purpose of this biochemical test? We know that the bacteria during their life cycle carry out a lot of biochemical processes. And when I give a particular substrate, if there is a biochemical reaction on the substrate, there is a resultant product that is produced. Either the product is colored or the product, when attached to some other strain, it will give a stain, it will give a color. Based on that, I will be able to know whether this particular bacteria is having that activity. Therefore, that particular bacteria is causing the disease. So here, the results that you are observing are either based on the change in color of the medium, because there is a change in the pH. That is, there is a product that is coming outcome of a particular chemical reaction. That product, when it is released into the broth, it will change the pH, resulting in the change of the color. Or I can either visually detect the bacterial growth with the help of essential nutrients. That means a particular nutrient is provided. For example, I'm doing a test like a particular sugar alcohol test, or I'm doing, let's say, a lactose broth test. Only those bacteria which have an ability to utilize this particular nutrient will be able to grow in the presence of that essential nutrient. So based on that, I will be able to detect what bacteria is growing in the given medium or in the given broth. So for biochemical tests, the important thing is the medium that you are taking, or the broth or the culture medium that you are taking to grow the bacteria in, so that you will be able to check what type of a compound is coming out or what type of a color change is happening. They're very inexpensive, very simple to perform. So you will be able to distinguish microorganisms even to the species level. I will be able to study. So only staining is not enough for identifying the bacteria. I also need to do these biochemical tests. There are a variety of tests that are available in microbiological testing things. When you go on identifying bacteria, you have a huge number of tests that can be performed. So here you can see a list of tests: gram-positive bacteria, starting from catalase test, blood agar test, CAM test, starch hydrolysis test, coagulase test. You can do many of them. When you're talking about gram-negative bacteria, you can do a sugar test, methyl red test, nitrate test, Siemens citrate agar test, urease test, SIM test. So these are some tests which are specifically designed to for gram-positive, gram-negative bacteria. So let's say I identified a sample, I made a smear, I did the basic differential staining, gram staining. I got to know whether it is gram-positive or gram-negative. Further, I want to know what type of a gram-positive bacteria it is. I can do any of these tests or combination of 2 or 3 different tests to confirm what bacteria it is. Similarly, I got to know it is gram-negative. I will do a combination of tests to check what type of a bacteria it is. Now, suppose I don't have so much of time and facilities to perform so many of those tests to confirm what bacteria it is, then what I can do is I will have a simple 4 tests that are commonly done. We call them as IMVIC tests. These are named after the initials of 4 tests. These are a series of 4 biochemical tests which will help us identify the bacterial species, especially when you're doing coliform, like our E. coli. Now, most of the times, even during COVID, we saw that there are a lot of water samples which were getting contaminated. Other than virus, there were so many other bacterial infections that became more prevalent during that time. So when they want to test different samples from different locations, instead of doing all the tests, you can do these 4 simple biochemical tests to confirm whether that particular sample is infected with coliform or no. So I stands for indole test, methyl red test, Vaux-Proscore test, and citrate test. So in this, the indole test and citrate test, they will be able to detect the ability of bacteria to produce specific enzymes, and they utilize specific nutrients. Whereas the methyl red test and the VP test, they will detect the final metabolic product that is produced by the bacteria, utilizing these specific nutrients that we are providing in the tests. So first we will start talking about the indole test. So this is a specific test which detects the ability of the organisms to produce indole as a metabolic product which utilizes tryptophan. Now, generally, we all know that these bacteria survive on a lot of compounds. Now, tryptophan is one amino acid that can be produced. It's a byproduct. It's a metabolic byproduct that is produced. So, the reagent that we use in this, we call it as a Kovacs indole reagent. Now, what they do is, these bacteria which are giving a positive result with the indole test, they have an enzyme that is produced called as a tryptophanase. Now, what is the purpose of this? This enzyme will help metabolize tryptophan, which converts this tryptophan into indole and pyruvic acid. It produces indole, pyruvic acid, and ammonia. So when the indole reagent is added to the medium with the bacterial culture, it produces indole, which then combines with an aldehyde, giving rise to a distinctive red color. So you can observe here, if that particular bacteria has an ability to produce indole, when added with the aldehyde reagent, it will give you a red color. So if that particular sample doesn't have this particular bacteria that has an tryptophanase, therefore it will not convert tryptophan to indole. Therefore, it will not give you a red color. So the positive color indicates that there is a pink to violet red ring that is formed depending upon the reagent that is used. So there are various aldehyde molecules that can be used. I can add cinnamaldehyde. So the broth is prepared with that, and then I will add one of these cinnamaldehyde. In fact, if you're using Kovacs indole reagent, this uses amyl alcohol and benzaldehyde. So various reagents can use all those. Now what we do is we add this culture, you add the test bacteria, they're cultured in this particular broth. You leave them for up to 24 to 48 hours. Now remember that most of these biochemical tests, you will be able to detect, the results are visualized only after 1 or 2 days, because you must provide that environment for the bacteria to grow in the given broth. Only then you will be able to visualize the colors. So then you will see positive. If that particular bacteria belonging to the that particular class which can produce a tryptophanase, only then you will be able to see that they will give this red color. Otherwise, if they are negative, they will not be able to give you that color. So what are the bacteria that are positive? So indole positive test you will get from bacteria like E. coli, Vibrio cholerae, Enterococcus. They all give a positive. Whereas the negative, most of your Klebsiella species, they're all negative to this because they don't basically have this enzyme. Even Pseudomonas gives a negative one for Klebsiella, indole test. Then staph also is negative for indole test. So E. coli, Vibrio cholerae, Enterococcus, these species give a positive color. Klebsiella, Pseudomonas, staph, they give a negative color. So simple, what you do, you take a broth containing tryptophan, then you add this particular broth, you allow the bacteria to grow in the broth. Suppose the bacteria has the tryptophanase enzyme, they will convert the tryptophan present in the medium into indole, which can further react with the aldehyde that is also provided in the broth, giving rise to a characteristic color. If they are negative, then you will not see that particular color in the bacteria. Then you have a methyl red test. Now, this is a test where you will be able to produce, test the bacteria that have an ability to produce mixed acids as metabolic end products of glucose metabolism. Now, understand that bacteria will undergo a fermentation pathway as a part of their glucose metabolism, and they convert pyruvic acid. Now, we all know that the pyruvic acid produced as an end product of glycolysis. After glycolysis, the pyruvic acid produced will have different fates. Either it can undergo aerobic respiration or it can undergo anaerobic respiration in the form of fermentation. So depending upon different bacteria, they will produce different types of acids. So glucose in glycolysis produces pyruvate. Pyruvate undergoes mixed acid fermentation, giving rise to different types of acid end products. Now, depending upon what end product is being released out, they will alter the pH of the medium. So sometimes what happens is, depending upon what acid is coming out of it, the pH is altered. It can be reduced to 4.4 or lower. So methyl red is basically an indicator which can detect the change in the pH. So if the end product of fermentation is producing acids, that will convert methyl red into, or that will change the pH. Then when we add methyl red, it will change that medium into red color. So you will see in a positive result, the medium is changed into red color. If suppose they are not producing any kind of an acids, then you will see that there is a negative result where there is a yellow color that is being produced. So depending upon what type of end products are produced in acid end products are produced, they will give you a red color. If they're not producing, they'll give you a yellow color. So for 24 hours, you have to incubate it in a specific broth called as MRVP broth, and you add a methyl red indicator to it. So the positive result is development of red color. Negative result is development of yellowish color. So what are the positive bacteria? Bacteria that give a positive test. Again, E. coli is positive to methyl red test. Salmonella bacteria are positive to methyl red. Even staph. Whereas bacteria that give negative color, you may have Klebsiella species, then Enterobacter. These will give a negative color with the methyl red test. A very interesting test that is further used is called as a Vogts-Proskauer test, or in short, we call it as a VP test. So this is also in that series of those 4 tests which detects the ability of bacteria to metabolize pyruvate into a neutral intermediate product called as acetyl methyl carbonyl or acetoin. So here you will see that generally in the tests, glucose gets converted into pyruvate. Now, pyruvate has various fates, as we just observed it. It can undergo many pathways in the process of trying to provide energy. So if it is entering into a butanediol pathway, an intermediate product that is produced is called as acetoin. It's also called as acetyl methyl carbinol. Now, when further it combines with a variety of reagents, like when I add alpha naphthol and potassium hydroxide, it will give rise to a pink red color. So in that bacteria, when they are specifically again grown in this particular broth, that is MRVP broth, for 48 hours, you allow it to have undergo aerobic respiration. Then you add these two reagents, one containing the oxygen and potassium hydroxide, then alpha naphthol. Then in 30 minutes, you will see that if that particular bacteria is able to convert this producing acetoin, then they combine with alpha naphthol and give rise to a pink red color, which indicates a positive test. In a negative test, you will see that there is no change in the color. That is a negative test. So VP test is exclusively used for the detection of production of acetoin. Now, sometimes what we do is, if suppose you know that the bacteria have a combination of these two, fermentation pathway you want to study, a combination of these two, that is a methyl red and VP test, together there is a test that is conducted that is called as an MRVP test. Instead of doing it separately, you do it together. MR turns it yellow and then VR turns it brownish green to yellow. Together also they can be performed. So sometimes the culture will only be positive for one. Either it is MR plus or VP plus, or it is sometimes you will see E. coli we said is positive to methyl red and negative to VP. So like this, you can make a list of depending upon what species it is and to which test it is giving positive or negative. So together MR and VP tests are conducted. Now the last one in the series that we are talking about, indole, methyl red, VI and C. Here in VI, Vogue's test is what is important. I is just used for the formation of an abbreviation. It doesn't as such have any importance. The last test is the citrate utilization test. Now it detects the ability of bacteria to use citrate as a sole source of energy. So what are you trying to do? You allow the bacteria to grow only in a medium which is containing that citrate. So those bacteria which have a citrate permease enzyme, where they will be able to accept the citrate from the outside, allow it to enter into the inside, convert it into oxalic acid, produce pyruvate and provide energy to the cells, will only be able to grow in that specific medium containing citrate. So you will see that the positive result is growth, where the color of the slant is changing from green to intense blue. So those bacteria that are able to grow in this slant only containing citrate as a sole source of energy, because they have specific enzymes that can utilize, open up the cell wall, allow that citrate to come inside, use that as a source of energy, and then they are able to grow. They will give you a blue colored complex, and the blue color is produced because of the addition of bromothymol blue, and the pH is maintained. So you will see a kind of a deep blue color that is produced in those bacteria that are positive to citrate utilization. If the bacteria is not able to grow in that, there is no change in the color of the slant. It still remains green in color. This indicates that the bacteria is not having this particular enzyme where they have an ability to utilize citrate. Now, these are the 4 tests. Commonly, those 4 tests along with the urease tests are used to rule out whether it is a coliform bacteria, Enterobacter, or any other species. Generally, to differentiate specifically if it is a Proteus vulgaris or if it is a not Enterobacter, we do this urease test. Now, urease is an enzyme that is produced by microorganisms. What do? Is they convert urea into ammonia. Now, whenever there is a release of ammonia, automatically there is a change in the pH. The medium turns alkaline because of the production of ammonia. Automatically, if the pH of the medium is changed, there is a change in the coloration. The phenol red indicator changes to deep pink color. Now, if the bacteria is not able to produce this urease, then there is no production of ammonia. So, therefore, there is no change in the color. So, we use a specific medium for this. We call it as a Christensen's urease agar medium, which you allow the bacteria to grow in this, and you add a phenol red indicator. So, what is happening? Those bacteria which we have confirmed using our 4 biochemical tests plus an urease test, we are sure whether it is a gram E. coli, or whether it is Enterobacter, whether it is staph, or whether it is Proteus, to confirm that. Sometimes we add an urease test also into that category of IMVIC tests. So what are the uses of this test? Generally, as we have been discussing, why do we do these biochemical tests? Very important. They're used in clinical research, teaching, in laboratories. Even if when you want to go up to the genus level and then identify what bacteria it is, we will do it. Now, as I said, along with urea and triple sugar iron tests, they will be able to further differentiate the enterobacterial species. So some tests, they're specific to differentiate the species in a genus. Like as I said, indole test is used to differentiate Klebsiella. Different species of Klebsiella can be identified. Similarly, in Proteus vulgaris, Proteus mirabilis, you will be able to differentiate them even to the species level in certain tests. But generally what happens is, they are not enough when you want to go to the species level. These tests are not enough. Further additional tests have to be performed, specifically when you want to identify. So I will just review some of these important tests where you can identify bacteria based upon their specific features. Like certain bacteria have an ability to reduce the nitrates that are present in the soil. Some bacteria have an ability to reduce the sulfur present in the samples. So based upon their ability to convert these substances into different products, you will be able to detect them. So for example, you have a nitrate reducing bacteria. They are very important in soil microbiology because they can recycle nitrogen. They will convert the nitrates into nitrites, and then depending on their ability, if they're producing a nitrate, it will give you a red color compound in the broth. If they're not able to convert, you don't see any red color. So positive samples like E. coli, they are nitrate positive, they will give you a color. Similarly, you see Pseudomonas, this is also nitrate positive, whereas Acinetobacter, it is nitrate negative. So depending upon that, you will be able to study that ability of the bacteria. Similarly, sulfur reduction test, there are some bacteria which have this ability to reduce sulfur. So sulfur reduction test is exclusively done to those, because the end product in this is a hydrogen sulfide gas. Now, this gas is detected by the indicator that you add during the test. So if there is a hydrogen sulfide that is produced, then you will see that there is a black precipitate form. If the hydrogen sulfide gas is not produced, there is no black precipitate form. So for this, you have specific medium that is produced. It's called as a sulfur indole motility medium, or a SIM medium, very prominently used to differentiate between your coliform bacteria and Enterobacter. You have other medium that are used, like triple sugar medium, lead acetate agar medium. They're all used to study the ability of the bacteria to reduce sulfur. Similarly, starch hydrolysis is done. This is where you will see that certain bacteria can hydrolyze starch because they have enzymes like alpha amylases and glucosidases. So you all know that starch is a polymer. It has both alpha 1,4 linked glucose residues, and also it has alpha 1,6 branch points. So certain bacteria which can utilize exclusively starch completely, they are able to utilize both the alpha 1,4 linked amylose and alpha 1,6 linked amylopectin, then they will be able to grow. So when you add iodine, what happens is, wherever the starch has been utilized, then they will form a dark brown color. Whereas those bacteria which are not able to carry out the hydrolysis of starch, they will create a clear zone around the bacterial growth, indicating that the bacteria did not utilize any starch. So starch hydrolysis test also is a useful one. Similarly, a catalase test is done. This is where, remember in the introduction, I said the purpose of bacterial biochemical tests for identifying them is their ability to use a particular enzyme and give rise to a product. So when we add hydrogen peroxide to the medium, you will see that the microorganisms will produce bubbles. So in a positive test, when we put a Durham tube, you will see that there are air bubbles produced if that particular bacteria is able to use hydrogen peroxide and produce oxygen. Whereas in negative test, you will see the H2O2 is not utilized because they don't have a catalase enzyme. There are no bubbles produced. Similarly, there is a hydrogen sulfide test we saw, same thing, sulfur-containing bacteria can produce this. Okay, so these are all different types of tests. If you start talking, there are variety of tests that are available for different microorganisms, and you can go up to the species level and you can identify. So what are the kind of questions that you can expect from staining and biochemical tests? When you're talking about staining, you will have simple questions that can be asked. That is the type of the stain, what is the primary stain, secondary stain, common questions. But when it comes to biochemical tests, the questions can be more critical. They can give you a kind of matching type of a questions. They will give you a list of stains and they will say which organisms are positive, which organisms are negative. So first you should know, you should be able to differentiate the microorganisms based upon their grouping, structure, characterization, and then you will be able to answer the question. So you can just look at some of the important questions. First, this is an interesting question that you can see. What is the correct order of staining reagents in gram staining? What do we add first? What is the primary stain? What is the next step? And what is the last stain? This is important. Now, depending upon their cell wall structure, some bacteria allow the antibiotics to enter and some don't. So based upon their cell wall structure, you can get some questions because you are differentiating them based upon their differential staining ability. So gram-positive bacteria are usually susceptible to which kind of antibiotics? Now you will get questions like which of these staining techniques helps in demonstrating the spore structure in bacteria as well as free spores. So as remember, I told you there are specific techniques like endospore staining exclusively used for detecting bacteria that have an ability to produce spores. What is the role of iodine in gram staining? I said what is a primary stain, what is a mordant, what is a secondary stain. So remember that. Then which bacteria appears purple violet color after staining? Very clear. What gram positive, what color is produced? Gram negative, what color is produced? White. Then lipopolysaccharide in cell walls is characteristic. So remember which bacteria has an LPS layer, which bacteria do not. I gave you a tabular column which says which is positive, which is negative to the bacteria. And which of the following is an example of gram-negative bacteria? Very important. You should know some examples at least for gram-negative and gram-positive bacteria. Then peptidoglycan layer is present in large quantity in which type of a bacteria? So let me show you this image where I gave you a differentiation in the cell wall structures. Please remember these two images very clearly. If you have this in your memory, I think when any question asked about the cell wall structure and this tabular column, gram positive has a thick peptidoglycan layer, whereas gram negative don't have. Gram negative has lipopolysaccharide layer, but gram positive doesn't have. Teichoic acid is an external thing present on the peptidoglycan layer only in gram positive, but not in gram negative. So this is an important slide for you to answer many questions that are asked related to staining techniques. Okay. Now, so again, gram-negative bacteria are more resistant to antibiotics. Why? Because they have a outer lipopolysaccharide layer, which acts as a very solid support. And they remember I told you they have porins. They don't allow anything to come in and come out very easily. So they have a permeability test check that is done before allowing anything inside and outside. Then which of the following are true for gram-negative bacteria? Understand what happens when this crystal violet iodine, that is CVI complex, is added. What happens to gram-negative bacteria? What happens to gram-positive? Here, let me explain. I think I already told you. When the gram-negative bacteria is added with this, the lipopolysaccharide layer is destabilized and therefore it doesn't retain any of the crystal violet or iodine. That is the reason why when you wash it and add saffron in, it takes up the saffron in color, but not the crystal violet color. Now, this kind of questions can also come where I said you will have matched the following. Now, the type of organism name is given and which tests. So, you should very clearly know gram-negative bacteria, E. coli, which test is positive. Remember, we said you should be able to identify which bacteria is MR positive, VP minus, IT positive, CT positive. Okay, that is whenever you are talking about microorganisms, make a tabular column like this, name of the organism and name of the test, all the 4 tests. Let's say you talk about indole test, methyl red test, Vogts test, citrate test. Then start making your organism name, that is E. coli. positive, negative, negative, positive, Klebsiella, Streptococcus, Staphylococcus, Pseudomonas. Like this, try to make a tabular column like this. So you will never forget. I mean, when it comes to entrance point of thing, this is this kind of a tabular column you should be able to make on your own. Read the tests, understand the principle, talk about the organism, and make a tabular column like this. Then you will be able to clearly answer this kind of a questions. Now, which test is mainly used for differentiation between Enterobacteriaceae from gram-negative bacteria? So remember I told you, apart from the IMVIC tests, we also do the other tests like a urease test where I want to differentiate from Enterobacteria from coliform bacteria. Then urease test, what is the medium? I said Kristen's urease agar medium is used. Now, Siemens citrate medium, where is it used? Because we are doing it a citrate test, their ability to test the citrate. So what do you observe in this? Both color and also growth. Both are observed or only one is observed. So you will see this kind of questions also you should be able to observe, understand the test clearly, and then you will be able to note this. Then acid fast staining is done for what type of a bacteria? Remember we said it is exclusively done for the bacteria belonging to the Mycobacterium species, or if you cannot even remember this, remember this is important for bacteria causing tuberculosis. Acid fast stain. Then acetoin, what did I say? This is a compound that is produced specifically in indole test. If that is converting this into acetoin, then it will give you a positive result. Now, this is about the fermentation. I said their ability to produce mixed acids is checked because of the change in the pH. What is the test that we discussed which detects the changes in the pH below 4.4? I said a methyl red test. So like this, everything about the test you should remember first. The critical points, broth used, conditions that are applied, end product, color change. So all these things you have to follow. I'll repeat. Remember the broth used, conditions applied, end products produced. If there is any specificity for a particular test, you should also know what is the test specific for. I mean, specificity to a particular organism or to a particular genus or to a particular species. Remember that. So if you are studying these tests from this perspective, I think you will be able to answer most of the questions in the entrance perspective. Okay. So you will have these kinds of questions anyway. Indole, what is the reagent that is used? So I said it's a Kovacs indole reagent. Now this kind of questions you will get. Choose the correct sequence of true or false for the following statements. Like which of these is true? So whenever you are reading these kinds of questions, so first you write down in your own form, read every sentence, write whether it is true, false, true, or what is it, and then identify which of these options suits best to your answer that you have already marked. Okay. So I'm not giving you any answers in this because I want all of you to take the test that is given, answer the questions, and then try to evaluate on your own. So before we close, a quick review. Staining, important because we want to highlight the organism against a background when you visualize it under a microscope. Simple stain, only one type of a color, one type of a stain is used and all bacteria will take the same color. Differential staining, different colors because depending upon the cell wall characteristic feature of a particular microorganism when it comes to gram staining or its ability to retain acid or not retain an acid color, acid or alcohol, that is in an acid fast staining. Special features, endospore staining, flagellar staining, negative staining, these are also can be performed. When it comes to biochemical tests, there are a huge list of tests that can be performed, but a series of 4 tests initially are done to rule out, I mean, to understand first whether it is an E. coli or whether it is an Enterobacter or whether it is a Proteus species or a Staph species. That is the indole test, methyl red test, Vogt's test, citrate test. Commonly done. For any of them, the procedure is prepare a medium, grow the bacteria in the medium. If the bacteria has an ability to convert any compound in that medium and give rise to a product or produce a gas or produce a pH change, Then it will be visualized either as a color change or production of bubbles or growth. You will be able to visualize it. Then you will be considering whether that particular bacteria is positive or negative in the test. Apart from that, various other tests depending upon the ability of the microorganism or the bacteria to convert nitrates, sulfur, hydrogen sulfur, hydrogen peroxide. You can do any of these tests to detect the bacteria. All this identification of bacteria is a crucial step for diagnostic purposes, for research laboratory studies, and also for general practical analysis also, because you are handling microorganisms, should know whether they are pathogenic, non-pathogenic. All these understanding can be done if you identify the bacteria, both by staining techniques and biochemical tests. So please make your own understanding. As I said, make a tabular column when you're doing biochemical tests, organisms, and positive, negative. So that will definitely help you answer any kind of a question in any kind of an examination. So good luck. Thank you.